there, and welcome to this video on GCSE Biology for the AQA Specification, focusing on the topic of animal tissues, organs and organ systems, and in particular focusing on the blood. I'm Shumina from StudyMind, where we help you revise GCSE Biology with our helpful video tutorials, tailored to your subject, your specification, and to you. If you're new here, make sure you click the subscribe button. Whilst you are watching, please leave any comments below if you're unsure about anything, and let us know if it's your first time watching our videos so we can send you our free revision materials. We also have helpful timestamps below for each part of the video to help guide you through the specification. So let's get started. Hello and welcome to tutorial 3 of 7 on organ systems, and today we're going to be having a look at blood. So in our last session we had a look at the structure of the heart and the roles of these different parts, and we also discussed the key blood vessels associated with the heart and the lungs. So do go back and have a look at that if you haven't seen it already. But in today's session we're going to be having a look at the following components of blood. So there's essentially 4 really important components of blood, and those are your white blood cells, your red blood cells, your platelets and your plasma. And notice how they all have really distinctive looks. So this isn't just for the purposes of this diagram, they do actually have distinctive structures, and so you can identify various components of the blood. So what we're going to do in today's session is go through these individual components and have a look at their structure and their function, because ultimately you want to be able to describe each of these and also to be able to identify them from a diagram. And as I just said, in your exam you might actually be presented with a diagram, so it's really important that you gauge the structural features of these types of cells. So first of all, let's have a look at plasma. So plasma is referring to the watery fluid where all the components of blood flow. So this means that it carries all the components of blood that we were talking about earlier on, so your platelets, your red and white blood cells, but also it carries carbon dioxide, glucose, amino acids, urea, hormones, antibodies, antitoxins and proteins. So anything that you can think of that's carried in the blood is essentially carried in the plasma, apart from oxygen, which is mainly carried in blood cells, red blood cells, but some oxygen may also be dissolved in plasma, but only a very small amount. So now let's come on to the structure of plasma. Well, plasma is clear and watery, and various things are transported to various places around the body via the plasma. So, for example, glucose is transported from the small intestine, where it's absorbed in the process of digestion, to either the body cells for respiration or to the liver for storage, because remember, glucose is stored as glycogen in the liver and also in muscle cells. And similarly, hormones are transported from endocrine organs to their target cells, also in the plasma. So you can see here how the plasma is a really important part of the transportation function of blood. So composition-wise, the majority of the blood is made up of the plasma, so around 60%, and the other 40% is made up of the other 3 components of blood that we're going to be looking at later on in this session. And just keep in mind that AQA often ask you to name constituents of the plasma, so do go back to that list that I went through earlier on and make sure that you commit that to memory. So we've looked at component number 1 of blood, which is the plasma. Now let's have a look at the red blood cells. So their function is to transport oxygen around the body. And, go back to that. So in the lungs, red blood cells associate with oxygen to form oxygen bound to a pigment in them called hemoglobin. And we refer to this complete structure as oxyhemoglobin because the oxygen has bound to the hemoglobin. So we combine those words together essentially and we form the word oxyhemoglobin. So that's in the lungs, and this is when deoxygenated blood passes through the lungs, picks up these oxygen molecules and becomes oxygenated. And then this blood will then get transferred towards tissues which are respiring. And here, the oxyhemoglobin is going to dissociate from the oxygen molecules. And so it's going to go back to form oxygen plus hemoglobin, and the oxygen will be released to these respiring tissues. And meanwhile, the hemoglobin is now deoxygenated, but it will still stay in the blood, and it will be returned to the lungs where it's going to get oxygenated again. So red blood cells, they transport oxygen around the body. They contain this pigment called hemoglobin, which binds to oxygen, forming oxyhemoglobin. And this oxyhemoglobin is then going to reach respiring tissues where it splits back into oxygen plus hemoglobin. So this sign here means that this reaction is reversible. So we can get oxygen and hemoglobin combining in the lungs to form oxyhemoglobin. And then in the respiring tissues, we're going to get oxyhemoglobin dissociating or splitting up to form hemoglobin plus oxygen. So it's a two-way thing. It can go this way or that way. That's all that sign means. So structure-wise, red blood cells have a biconcave disc shape. And this is really, really important because it increases their surface area. And so therefore they've got a higher surface area for the uptake and release of oxygen, which is essential to their function. Red blood cells also have no nucleus, so this means that they have even more space to pack in as much hemoglobin as possible, which is going to make them really, really efficient transporters of oxygen. So if an exam question ever asks you to relate the structure to the function, just always sit back in your chair and think, right, so I've got a red blood cell, I wonder how its structure is related to its function. Well, I know that red blood cells, their function is to carry oxygen around the body, so how is their structure related to this? And then hopefully that will trigger your memory of it having a biconcave disc shape, which increases the surface area for oxygen uptake, but also oxygen release. Also, red blood cells have no nucleus, so they can pack more hemoglobin in, which means that they've got more carrying capacity for oxygen. So just always relate the structure of the cell, or whatever we're talking about, to the function. And one more point to make here is that most cells do have a nucleus. So red blood cells are one of the very few cells in our human body that do not have a nucleus. So this slide just summarises what I just said on the previous slide about the adaptations of the red blood cell to its function. So remember the biconcave disc shape and also no nucleus. So we've had a look at plasma and red blood cells, so now let's have a look at white blood cells. So white blood cells are completely separate entities to red blood cells, and they help protect against infection and foreign pathogens. So they're essentially the police force of our body. So there are two types of white blood cell, phagocytes and lymphocytes. And phagocytes make up the majority of white blood cells, so around 75% of white blood cells are phagocytes, but meanwhile lymphocytes make up the other 25%. So let's take a look into their function. So remember, they're both going to be protective to the body somehow. So phagocytes form a defence system by essentially eating your pathogen. So phagocytes detect that there is a bacterium, for example, in the body, but remember a pathogen can be multiple other things, such as viruses too. And so the phagocytes detected the presence of a pathogen, perhaps by recognising an antigen on its surface, and it's going to move towards the pathogen and it will literally engulf it. So if I were to draw what would happen next, it would, oops, this isn't drawing, oh no. Oh, there we are. No. And there we are. So this is your phagocyte here. Let me redraw that. My pen's losing connection. So this is your phagocyte here, and it's literally going to move round and engulf your pathogen. Okay? So it literally eats your pathogen, and that's how it contributes to defence in the human body. But also, we have lymphocytes. Now these produce defence via a different mechanism. So lymphocytes produce antibodies to fight against pathogens, and we have explored this in a different tutorial, so do go back and find that tutorial on immunity, where we look at this in a lot more detail. But in terms of the function of white blood cells, this is all you need to know. So there's phagocytes and lymphocytes. Phagocytes engulf or phagocytose pathogens, basically eating them, whereas lymphocytes produce antibodies that fight against pathogens. And so how is the structure of white blood cells related to their function? Well, they have flexible cell membranes which form pseudopodia, which basically means that white blood cells are able to engulf pathogens. Also, white blood cells have a lobed nucleus, so look at this horseshoe shape of its nucleus. So that's how you're going to identify a white blood cell, for example, in an exam. So the diagram will have a horse-shaped nucleus in the cell. And also there are these structures called lysozymes, which contain digestive enzymes called lysozymes. So they contain enzymes known as lysozymes, which are your digestive enzymes. So the flexible cell membrane, the lysozyme enzymes in the cytoplasm, which help in digesting foreign pathogens, and also the lobed nucleus. Now let's move on to platelets. So the function of platelets are to clot the blood and reduce blood flow around cuts and wounds. So if you have too many platelets, you're going to get a lot of clotting, and so you're going to be really prone to blood clots. If you have too few platelets, you're not going to be very good at clotting at all, which can lead to problems. So if you get a simple paper cut, for example, you're going to bleed a lot more than a person who has a normal content of platelets in their blood. So let's look at the structure of platelets. So they're essentially fragments of cells, so they have no nucleus. But they do have cell surface proteins, which allow them to clump up together and close up a broken blood vessel. So this is how they clot, they form a clot. They also have stored proteins, and basically these stored proteins are released when there's any damage to endothelium, for example, and this initiates a sequence of reactions. A clotting cascade is what we call it, and therefore initiates formation of a clot. So today, just to summarise, we had a look at the four main constituents of blood and broke them down into their structure and function. And it's really, really important that you understand this well, because in your exam you may be presented with an image you've not seen before, and you may be expected to label what type of cell is present. So well done for today, and I'll see you for the next session.
Thanks for watching this free video from StudyMind. If you liked this video, make sure to subscribe to catch our newest videos by clicking below, and leave a comment on a topic you'd like a video on. Click here to watch more videos in our series for GCSE Biology, or visit our website studymind.co.uk for free past paper compilations by topic and specification.